Lecture Notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046104557. 4. Cell membranes and transport. Cell membrane is a phospholipid bilayer that regulates the entry and exit of molecules. Diffusion, osmosis, and active transport are some forms of transport seen across the cell membrane. 4.1 Fluid mosaic membranes. Fluid means molecules of membrane move about, as if membrane flowing. Basic membrane structure consists of bilipid layer of phospholipids and protein molecules are moving through it. Certain other molecules also move through it, i.e. by diffusion. Mosaic means protein molecules interspersed or scattered throughout the fluid layer of phospholipids. Proteins are of wide variety like enzymes, hormones, structural proteins, etc. The cell surface membrane controls exchange between the cell and its environment. Some chemical reactions like place on membranes inside cell organelles, as in photosynthesis and respiration. 4.1-1 Description of fluid mosaic model of membrane. The fluid mosaic model describes the structure of the plasma membrane as a mosaic of components, including phospholipids, cholesterol, proteins, and carbohydrates, that gives the membrane a fluid character. Plasma membranes range from 5 to 10 nanometers in thickness. 4.1-2 Description of arrangement of cholesterol, glycolipids, and glycoproteins in cell membrane. The basic structure of a membrane is a 7 nanometers thick phospholipid bilayer containing protein molecules. The structure is described as a fluid mosaic. 4.1-3 Roles of phospholipids, cholesterol, glycolipids, proteins, and glycoproteins. Phospholipid bilayers are a barrier to most water-soluble substances because the interior of the membrane is hydrophobic. Cholesterol is also needed for membrane fluidity and stability. Some proteins are transport proteins, transporting molecules or ions across the membrane. They may be either channel proteins or carrier proteins. Channel proteins have a fixed shape, carrier proteins change shape. Some proteins act as enzymes. Glycolipids and glycoproteins form receptors, for example, for hormones or neurotransmitters. Glycolipids and glycoproteins also form cell-cell recognition markers. 4.1-4 Main stages in the process of cell signaling. Membranes play an important role in cell signaling, the means by which cells communicate with each other. Cell signaling, the molecular mechanisms by which cells detect and respond to external stimuli, including communication between cells. The process that helps body cells communicate with internal/external environment and with other cells is called cell signaling. It is a kind of communication network to intercommunicative cells within the body. Lecture Notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046104557. Glycoproteins and glycolipids act as receptors. Different cells have different receptors. 3 major groups of receptors. 1. Signaling receptors reach messenger molecules such as neurotransmitters, hormones, etc. One good example of receptors is glucagon in the liver cells. The cells that lack glucagon receptors are immune to glucagon. 2. Receptors are involved in endocytosis. They bind to molecules that are parts of the structures to be engulfed by the cell surface membrane, e.g. phagocytosis. 3. Cell adhesion, involved in binding cells to other cells in tissues and organs of animals. Other membrane proteins may be enzymes that begins the signal transduction cascade, for example adenylate cyclase, a membrane-bound enzyme activated by G protein. The signal binds to specific receptors, and this spans the membrane. Changing the shape of the receptors allows it to interact with G protein, activate the enzyme adenylate cyclase, which in turn make AMP, second message, triggered the sequence of events called signaling cascade. Such type of pathway involved hormones glucagon or adrenaline to release glucose in blood depending upon requirement of body. Membrane system: cell surface membrane found in both animal and plant cells. Structure: the outer boundary of each cell consists of a surface membrane that is partially permeable. It is only about 7 nanometers thick. It is only visible under the electron microscope. It has a fluid mosaic model because of the arrangement of proteins that float in the phospholipid bilayer in a mosaic pattern, which is also capable of movement, hence it is fluid in nature. Lipids and proteins in the cell surface membrane can also rotate on their own axis, therefore this contributes to the fluid. Functions: 1. Serves as a regulatory barrier to control the transport of substances in as well as out of the cell. 2. The hydrophobic and hydrophilic portions control the entry and exit of fat-soluble and water-soluble molecules respectively, therefore they help to maintain ion concentrations. 3. Provides shape and the proteins give structural support to the cell. 4. Cholesterol prevents the phospholipids from packing tightly together, thus the cell surface membrane is more flexible and. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. Description: the proteins are held in the membrane because they are amphoteric, have both basic and acidic properties, as proteins are zwitterions, having positively and negatively charges groups. This enables the proteins to interact and be part of the bilayer membrane of phospholipids. The channel surfaces of the proteins are hydrophilic to enable ions to pass through. Next to the phospholipid tails, the surfaces of the proteins are hydrophobic to enable the protein to stay in the membrane by interacting with the hydrophobic hydrocarbon tails. Cholesterol is found embedded in the phospholipid bilayer. Stable. 5. Maintain the ionic balance in surroundings via active transport process through sodium-potassium pumps, which removes sodium ions from cells while actively accumulating potassium ions into them from their surroundings. 6. Proteins transport water-soluble ions across the membrane by the process of facilitated diffusion by forming channels so that ions are able to pass through the cell membrane. 7. Glycoproteins function as antigens to allow cells to be recognized by other agents, e.g. enzymes, hormones and antibodies. 8. Carbohydrate chains, glycolipids, attached to the surface of the membrane act as recognition sites, e.g. the human ABO blood system is the result of different glycolipids on the cell membrane of the red blood cells. 4.2 Movement into and out of cells. Diffusion, osmosis and active transport are three main ways that molecules and ions can move into and out of cells through the membrane. 4.2-1 Description of simple diffusion, facilitated diffusion, osmosis, active transport, endocytosis and exocytosis. Substances may enter or leave cells by diffusion, osmosis, facilitated diffusion and active transport. This process requires energy in the form of ATP. Water moves from regions of higher water potential to regions of lower water potential. When water moves from regions of higher water potential to regions of lower water potential through a partially permeable membrane, such as the cell surface membrane, this diffusion of water is called osmosis. Comparing diffusion, facilitated diffusion and active transport. Simple diffusion, facilitated diffusion, active transport type of membrane molecule involve lipids, proteins. Force driving the process, concentration gradient, ATP hydrolysis. Direction of transport with concentration gradient against concentration gradient, specificity nonspecific, specific saturation at high concentration of transported molecules. No, yes. Diffusion is the net movement of molecules or ions from a region of their higher concentration to one of lower concentration. Oxygen, carbon dioxide and water cross membranes by diffusion. Diffusion of ions and polar molecules through membranes is allowed by transport proteins. This process is called facilitated diffusion. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. Diffusion, facilitated diffusion and osmosis depend on the energy inherent in molecules and ions. Because the cell does not use energy in these processes, they are referred to as passive transport. Diffusion is sometimes helped by specific proteins that form part of the structure of membrane. This process is called facilitated diffusion. 4.2-2 Investigation of simple diffusion and osmosis by using plant tissues and non-living membranes including dialysis membrane and agar as found in nature. Agar is a mixture of two components, the linear polysaccharide agarose and a heterogeneous mixture of smaller molecules called agaropectin. Agar plate agar, disambiguation. Agarose gel electrophoresis. 4.2-3 Illustration of the surface area to volume ratio. Water enters the plant through root hairs by osmosis. Water crosses the root either through the apoplast pathway or the symplast pathway. It enters the xylem vessels at the center of the root. The main gaseous exchange surfaces in dicotyledonous plants are the spongy mesophyll layer and stomata in leaves. Gaseous exchange also occurs in the root hair cells and lenticels in bark. 4.2-4 Effect of changing surface area to volume ratio on diffusion by using agar blocks of different sizes. When an agar cube, or biological cell/organism, has more volume but proportionately less surface area, diffusion takes longer and is less effective. In more precise scientific terms, the greater the surface area to volume ratio, the faster the rate of diffusion. 4.2-5 Investigation of the fact of immersing plant tissue in solutions of different water potentials. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. Plant cells have cell walls. This is quite important when thinking about the effect of immersing plant tissue in solutions of different concentrations. Cells are primarily made of water. On average, about 70% of total cell mass is water. Every cell's cytoplasm has its own specific concentration of solutes, and this concentration is usually pretty similar across the same type of tissue, e.g. palisade cells will have similar concentrations of solutes in their cell cytoplasm, and that the pressure that water applies in plants, i.e. the water pressure, is known as turgor pressure. Turgidity is the state of being turgid or swollen, especially due to high fluid content. Plants need turgid cells to help them maintain their shape and in turn, help the plant stay upright. Water is mainly stored in the vacuole in the cytoplasm, and it is mainly this vacuole that regulates the turgidity of a plant cell. When you immerse plant tissue in solutions of lower water potential, hypertonic solution, than that of the plant cells, water diffuses out of the cell by osmosis. This means there is less matter inside the cell. This causes the cytoplasm to shrink, and thus the cell membrane gets ripped away from the cell wall. This process is called plasmolysis. Cells become weak and flaccid, as there isn't enough cytoplasm to support the cell and help it maintain its shape. When you immerse plant tissue in a solution of equal water potential to their cell cytoplasm, isotonic solution. Since the concentration of the solution is equal inside and outside of the plant cells, there is no net movement of water. This means the volume or shape of the plant cell is unlikely to change. When you immerse plant tissue in solutions of higher water potential than their cell cytoplasm, hypotonic solution. Here, the solution inside the cells is more concentrated than solution outside, so water diffuses down its concentration gradient into the cell, by osmosis. This causes the amount of cell matter inside the cell to increase. As the cytoplasm enlarges, it pushes outwards on the cell surface membrane more and more. Normally, this would usually cause the cell surface membrane to eventually burst, once the pressure, otherwise known as turgor pressure, in this case, grows too large. However, plant cells have very strong cell walls. This holds the plant cell intact, and as the cytoplasm pushes outside, the cell simply swells to its full size and becomes rigid. This cell is turgid. Water potential is the potential of water to leave a system. This is affected by water pressure, the volume of the water relative to the volume of the system, e.g. a lot of water in a small system will force water out of the system, and the concentration of the water. Osmosis is the net movement of water molecules from a region of higher water. Potential, dilute solution, to a region of lower water potential, concentrated. Solution, through a partially permeable membrane. In the uptake of water by plants, the soil around the plant must contain water that is more dilute than the water in root hair cells. This allows water to diffuse in by osmosis. The water outside the root hair cell will have a higher water potential than the water inside the root hair cell. The importance of water potential and osmosis on animal cells and tissues. Unlike plant cells, animal cells do not have a cell wall to support them. So, if you insert them in a hypertonic solution, water will move out of the cell by osmosis, causing the cell to shrivel up. If you immerse them in an isotonic solution, there will be no net movement of water. If you immerse them in a hypotonic solution, the water will diffuse into the animal cell by osmosis. However, there is no cell wall to keep the cell intact, so the water pressure grows so great that the cell bursts. It undergoes lysis. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. 4.2 to 6 Movement of water between cells and solutions in terms of water potentials. Water potential is the ability of a solution to absorb water molecules by osmosis. It is measured in units of pressure called kilopascals, kPa. Pure water has a water potential of 0 kilopascals. A solution always has a negative water potential, e.g., 0 kilopascals. Osmosis is the diffusion of water. It is the net movement of water through a partially permeable membrane from a solution of less negative water potential to a solution of more negative water potential. Effects of osmosis on a cell: tonicity of solution concentrations, net movement of water, effect on cell: solute water isotonic, same as cell none hypotonic, less than cell more than cell gains water, swells, turgor pressure hypertonic, more than cell less than cell loses water, shrinks, plasmolysis. Adding solute lowers the water potential. Adding pressure to a solution increases the water potential. In dilute solutions, animal cells burst as water moves into the cytoplasm from the solution. In dilute solutions, a plant cell does not burst, because the cell wall provides resistance to prevent it expanding. In concentrated solutions, animal cells shrink, while in plant cells the protoplast shrinks away from the cell wall in a process known as plasmolysis. Some ions and molecules move across membranes by active transport against the concentration gradient. This needs a carrier protein and ATP to provide energy. Exocytosis and endocytosis involves the formation of vacuoles to move larger quantities of materials respectively out of, or into, cells by bulk transport. There are two types of endocytosis, namely phagocytosis, cell eating, and pinocytosis, cell drinking. Getting in and out of cells. The membrane surrounding a cell is called the plasma membrane. The fluid mosaic model of membrane structure shows that membranes have a phospholipid bilayer. This bilayer is studded with proteins and polysaccharides. Proteins in the plasma membrane perform a variety of functions. They act as cell receptors, as recognition sites, and as adhesion sites. Proteins also assist the movement of molecules or ions through membranes. Diffusion is the net movement of molecules or ions down a concentration gradient. According to Fick's law, diffusion across exchange surfaces is faster when the exchange surface has a large area, the exchange surface is thin, and the concentration gradient is steep. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. Substances may cross the plasma membrane to enter or leave cells by 4 mechanisms. Mechanism description: ATP required example diffusion movement of molecules from a region of high concentration to low concentration. No oxygen, carbon dioxide, water, solid, liquid and gases, etc. Osmosis: diffusion of water from a high to low water potential through a differentially permeable membrane. No water. Facilitated diffusion movement of substances by attachment to transport proteins. No glucose. Active transport movement of substances against their concentration gradient, i.e. from lower to higher concentration. Yes, carrier proteins are also required: glucose, sugar, amino acids and ions, etc. Transport facilitated toward lesser concentration carrier sugars and amino acids. Endocytosis, phagocytosis toward inside vacuole formation cells and subcellular material. Pinocytosis toward inside vesicle formation macromolecules. Receptor mediated toward inside vesicle formation macromolecules. Exocytosis toward outside vesicle fuses with plasma membrane macromolecules. Investigations about diffusion and osmosis in animals and plant cells placed at different concentrations. The membranes that surround eukaryotic cells and the membranes that form their organelles all have the same basic structure, known as the fluid mosaic model. It is 7.5 nanometers thick and consists of a lipid bilayer with some proteins embedded in the surface and some running right through it. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. The interaction between the hydrophobic and hydrophilic ends of the phospholipids gives the membrane stability. Lipids also give the membrane selective permeability. Lipid-soluble, hydrophobic, molecules diffuse through the membrane easily. Hydrophilic substances cross the membrane through channels in the intrinsic proteins. The lipids and proteins can move laterally or change places, and this gives the membrane fluidity. This is essential for processes such as endocytosis, movement of material into a cell. A membrane is a phospholipid bilayer studded with proteins, polysaccharides and other sorts of lipid. This patchwork of molecules is like a mosaic. Because the molecules move about within their respective layer, the membrane behaves like a fluid. This is why the model is called the fluid mosaic model. The cell membrane is used to recognize chemical messengers, e.g. control the movement of substances into the cell, e.g. glucose, and release substances that cells have made, e.g. insulin. The proteins in membranes are of special interest to us. This is because they have a number of important functions. Proteins are carriers for water-soluble molecules, such as glucose. Proteins are channels for ions, such as sodium and chloride ions. Proteins are pumps which use energy to move water-soluble molecules and ions. Proteins are receptors, which enable hormones and nerve transmitters to bind to specific cells. Proteins are recognition sites which identify a cell as being of a particular type. Proteins are enzymes which speed up chemical reactions at the edge of the membrane. Proteins are adhesion sites which help some cells to stick together. Water potential is defined as the potential energy of water in the system to pure water when the pressure and temperature are held constant. The ability of water molecules to flow freely inside a given environment or system can also be measured using water potential. Solute potential, SIS, also called osmotic potential, is negative in a plant cell and 0 in distilled water. Typical values for cell cytoplasm are -0.5 to -1.0 MPa. Solutes reduce water potential, resulting in a negative psi-W, by consuming some of the potential energy available in the water. The kinetic energy of water is called water potential. Water potential reduces when a solute is dissolved in it. The magnitude of lowering of water potential because of solute is called solute potential. Water potential, psi equals psi, is the ability of a solution to absorb water molecules by osmosis. It is measured in units of pressure called kilopascals, kPa. Pure water has a water potential of 0 kilopascals. A solution always has a negative water potential, e.g., 6 kilopascals. Solute potential. The presence of solute molecules in a solution lowers its water potential. The greater the concentration of solutes, the lower, more negative, is the water potential. This change in water potential as a consequence of the presence of solute molecules is called solute potential, psi-s. As the solute molecules always lower the water potential, the value of the solute potential is always negative. Pressure potential. Take a situation such as that where water is entering a solution by osmosis. If a pressure were applied to the solution, then this would resist the entry of water. This pressure is known as the pressure potential and is given the symbol psi-p. Pressure potential is of importance in the water relations of plant cells as the cell wall can exert an influence. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. Key words: active transport, antigen, carrier protein, cell signaling, cell surface receptor, channel proteins, cholesterol, diffusion, endocytosis, exocytosis, facilitated diffusion, fluid mosaic model, glycoprotein, glycolipid, ligand, osmosis, passive transport, phospholipid, phospholipid bilayer, surface area to volume ratio, Visking tubing, water potential. Chapter Glossary. Active transport is the active movement of substances from a low concentration to a higher concentration, up their concentration gradient, with the use of energy in the form of ATP. Antigen is marker molecule on the cell surface membrane, usually a protein or glycoprotein, that can be detected by antibodies and triggers an immune response. Carrier protein is the protein involved in active transport that uses energy in the form of ATP to change conformation. Cell signaling are cells release chemicals which bind to complementary receptors on their target and trigger specific responses. Cell surface receptor is a component on the cell membrane which binds to extracellular signals. Channel proteins are transmembrane proteins for transporting large or charged substances. Some are involved in facilitated diffusion and do not use ATP, others are involved in active transport and do require energy in the form of ATP. Cholesterol is a steroid hormone which adds stability to the lipid bilayer. Diffusion is the passive spreading out of substances from a high concentration to a lower concentration, down their concentration gradient, without the use of energy. Endocytosis is a method of bulk transport into a cell which relies on invagination of the cell membrane and requires energy in the form of ATP. Exocytosis is a method of bulk transport out of a cell which occurs when vesicles fuse with the cell membrane and release their contents. It requires energy in the form of ATP. Facilitated diffusion is the passive movement of substances from a high concentration to a lower concentration, down their concentration gradient, through transport proteins without the use of energy. Fluid mosaic model is a model that describes membrane structure as a sea of mobile phospholipids studded with various proteins. Glycolipid is a lipid which is bound to a monosaccharide or oligosaccharide. Glycoprotein is a protein which is bound to a carbohydrate chain. Ligand is an ion or molecule that transmits signals within or between cells. It has a role in cellular signaling and recognition. Osmosis is the passive diffusion of water molecules from a region of high water potential to a region of lower water potential, down a water potential gradient, through a selectively permeable membrane without the use of energy. Passive transport is the movement of substances without the use of energy. Phospholipid is the type of lipid which forms the cell surface membrane bilayer. It is formed by the condensation of one molecule of glycerol, two molecules of fatty acid and a phosphate group. The two fatty acid chains are the nonpolar hydrophobic tails, whilst the phosphate group is the polar hydrophilic head. Phospholipid bilayer is a polar membrane made of two layers of phospholipid molecules. It is a selectively permeable barrier to the passage of ions and molecules into and out of cells. Lecture notes for AS Level Biology by Mohammad Rashid Chattha. Cell number 030046100457. Surface area to volume ratio is the volume of an object compared with the amount of area where it contacts its environment. Calculated by dividing the surface area of an object by its volume. Visking tubing is a semi-permeable material which can be used to model the cell surface membrane. Water potential is a measure of the tendency of water molecules to move from one area to another measured in kilopascals, kPa, and given the symbol psi. Short MCQs with right answers. 1. Which statement about cell signaling is correct? The binding of a ligand may cause a change to the shape of the receptor. 2. For students A, B, C and D, observed plant epidermal cells that had been placed in a concentrated sucrose solution for 30 minutes. They were asked to identify the partially permeable layer and to explain the appearance of the cells in terms of water potential and movement of water. Which student is correct? 3. 15. Which molecules in cell surface membranes are typically involved in cell recognition? 4. 16. What can increase the fluidity of the cell surface membrane? Cholesterol. 5. 17. The three main factors that affect the rate of diffusion across a membrane can be expressed by the relationship shown. 6. Which changes in the factors would result in the rate of diffusion doubling? 1. Surface area has doubled. 2. Thickness of membrane has halved. 7. 18. A student measured the time taken for complete diffusion of a dye into agar blocks of different sizes which were suspended in the dye. The results are shown. 8. What is the predicted time for complete diffusion of the dye into the agar block measuring 5 mm times 10 mm times 15 mm? 6.2s9. An experiment was carried out to investigate the effect of concentration of sucrose solution on cells in a plant tissue. A sample of plant tissue was cut into 7 cylinders of. Lecture notes for AS Level Biology by Mohammad Rashid Chattha. Cell number 030046100457. Equal length and diameter. The mass of each cylinder was recorded. Each of the 7 cylinders was put into a different sucrose solution concentration. 10. After 2 hours, the cylinders were removed, blotted dry and reweighed. The percentage change in mass of each cylinder was recorded. The graph shows the results of this investigation. 11. Which row explains the results if plant tissue cells were put in a sucrose solution of 0.45 mol and 3? 12. Which row shows the distribution of cholesterol and the carbohydrate chains of glycolipids and glycoproteins in a cell surface membrane? 13. Which row correctly describes all the possible relative concentrations of a substance when the substance is moved by endocytosis or exocytosis? Lecture notes for AS Level Biology by Mohammad Rashid Chattha. Cell number 030046100457. 14. Which statement about simple diffusion is correct? It is a passive mode of transporting substances. 15. Plant cells with the same water potential in their cytoplasm were each put into one of 3 different concentrations of sugar solution, 10%, 5% and 2.5%. The cells were left for 50 minutes and then observed using a light microscope. 16. Which statements are correct? 1. Cell Y had a lower water potential than the sugar solution it was put into. 2. cell Z was put into the 10% sugar solution. 3. Cell Z had a less negative water potential than the sugar solution it was put into. 17. Which of these substances can pass directly through cell surface membranes without using a carrier protein or a channel protein? CO2. 18. What happens to the surface area to volume ratio of a cube when the length of each side is doubled? The ratio halves. 19. Which roles of the cell surface membrane result from the properties of the phospholipids? 1. To allow cytokinesis to occur in mitotic cell division. 2. To allow entry and exit of oxygen and carbon dioxide. 3. To allow the phagocytosis of a bacterium into a cell. 20. The cell surface membrane structure is described as a fluid mosaic. Which statement describes the mosaic part of the cell surface membrane? The scattering of the different proteins within the phospholipid bilayer. 21. What describes the fluid mosaic model of membrane structure? Proteins and phospholipids move and change places. 22. A decrease in some factors will increase the fluidity of the cell surface membrane. Which pair of factors, when decreased, will increase the fluidity of the cell surface membrane? A decrease in 1. Proportion of phospholipids with saturated fatty acid chains. 2. Proportion of long fatty acid chains. 23. Which description of cell surface membrane permeability is correct? Without the presence of carrier and channel membrane proteins, the cell surface membrane has a low permeability to large polar molecules. 24. Which statements about the proteins and glycoproteins in cell surface membranes are correct? 1. They can allow cells to bond together to form tissues. 2. They can recognize messenger molecules like hormones. 3. They can be antigens and allow cell-to-cell recognition. 25. Which substances can pass directly through cell surface membranes and do not use a carrier protein or channel protein? CO2. Only 26. In plants adapted to cold conditions, their cell surface membranes change as the weather gets colder, allowing the plants to carry out exocytosis. Which change occurs in their cell surface membranes? An increase in the ratio of unsaturated phospholipids to saturated phospholipids. 27. Which statements about the cell surface membrane are correct? 1. Channel proteins allow water-soluble ions and molecules across the membrane. 2. Glucose can pass into the cell via carrier proteins. 3. Oxygen passes freely through the membrane as it is soluble in lipids. 4. Some glycoproteins act as antigens. 28. Cholesterol is an integral component of the cell surface membrane. Which statement about cholesterol is correct? It helps to regulate the fluidity of the cell surface membrane. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. 29. Which type of bond in phospholipids has a role in increasing the fluidity of cell surface membranes? C equals C30. Which of these substances can pass directly through cell surface membranes without using a carrier protein or a channel protein? CO2. Only 31. Which row shows a correct function of the components of the cell surface membrane? Phospholipids regulate the fluidity of the cell membrane. Cholesterol stabilizes the membrane over a range of temperatures. 3. Proteins form channels for polar molecules. 4. Glycoproteins allow cell-to-cell recognition. 32. Which functions are correct for components of the cell surface membrane? Stabilizing the hydrophobic layer, cholesterol, allowing osmosis to occur, phospholipids and proteins, barrier to dissolved ions, phospholipids. 33. The cell surface membrane structure is described as a fluid mosaic. Which statement describes the mosaic part of the cell surface membrane? The scattering of the different proteins within the phospholipid bilayer. 34. What describes a carrier protein in cell surface membranes? A glycoprotein that is involved in moving substances through the membranes by both active and passive transport. 35. What is the correct range of measurements for the width of the cell surface membrane? 5 to 10 nanometers. 36. Which molecules, found in cell surfaces membranes, help the immune system to identify cell? 1. Glycolipids. 2. Glycoproteins. 3. Proteins. 37. Which substances can pass directly through cell surface membranes without using a carrier protein or channel protein? Ca2+ and Na+. 38. Which molecules are found at the outer surface of a cell surface membrane? Cholesterol, absent; glycolipids, present; phospholipids, present. 39. What is a function of the cholesterol in a cell surface membrane? Regulating the flexibility of the membrane. 40. Which statements about the fluid mosaic model of a membrane are correct? 1. Glycoproteins in the outer layer of the membrane can move. 2. Channel proteins are fixed in position. 41. Which set of factors will produce the least fluid cell surface membrane? 1. Proportion of long fatty acid chains. 2. Proportion of phospholipids with saturated fatty acid chains. 42. Which set of factors will produce the most fluid cell surface membrane? 1. Proportion of long fatty acid chains. 2. Temperature. 3. Proportion of phospholipids with saturated fatty acid chains. 43. Membrane proteins are called peripheral if they are temporarily attached to the membrane, or integral if they are permanently attached to the membrane. Integral proteins are described as intrinsic if they extend across the whole bilayer and extrinsic if they are found only in one side of the bilayer. How would you describe a channel protein? Integral intrinsic. 44. Which two substances maintain the fluidity of the cell membranes? Phospholipid and cholesterol. 45. Some functions of molecules found in cell surface membranes are listed. Which are functions of cholesterol? 1. Maintaining fluidity. 2. Mechanical stability. 46. What is the width of the cell surface membrane? 5 to 10 nanometers. 47. In plants adapted to cold conditions, their cell surface membranes change as the weather gets colder, allowing the plants to carry out exocytosis. Which change occurs in their cell? Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. Surface membranes. An increase in the ratio of unsaturated phospholipids to saturated phospholipids. 48. Increasing which type of bond helps to increase the fluidity of the cell surface membrane? C = C49. One type of antigen is formed by a reaction between two different molecules. Apart from oxygen, which other elements are found in this antigen? Carbon, hydrogen, nitrogen, and sulfur. 50. Which molecules in cell surface membranes contribute to cell recognition? Glycolipids, glycoproteins, proteins. 51. The fluidity of the cell surface membrane can be changed by a number of factors. As the fluidity of cell surface membranes decreases, which process would be least changed? Active transport. 52. Which statement concerning the role of cholesterol in the cell surface membrane is correct? It maintains the fluidity of the membrane at low temperatures. 53. Which statement about the fluid mosaic model of membrane structure is correct? The more unsaturated the fatty acid chains of the phospholipids, the more fluid the membrane. 54. Which is correct for components of a cell surface membrane? Increases membrane fluidity. Cholesterol allows recognition of cell, glycoproteins. 55. Which molecules found in cell surface membranes contribute to cell recognition? 1. Glycolipids. 2. Glycoproteins. 56. Which of the following can decrease the fluidity of the cell surface membrane? 1. Fatty acids having longer chains 57. Which of the following can increase the fluidity of the cell surface membrane? Cholesterol 58. An increase in which component would make the cell surface membrane more fluid? Cholesterol 59. What is the role of cholesterol in the cell surface membrane? To regulate fluidity of the membrane 60. A student wrote 3 statements about cell signaling. Which statements are correct? Binding the signaling molecule 61. Which features are required to allow for efficient diffusion? 1. A large surface area 2. A short diffusion pathway 3. Maintenance of a constant diffusion gradient. 62. The following are all processes that allow movement into cells. Which processes require? ATP. 1. Phagocytosis 2. Active transport 63. When animal cells are cultured, salt solution is added to keep the cells alive. What is the purpose of the salt solution? To prevent net movement of water into or out of the cells 64. Liver cells contain vesicles that have proteins in their membranes which are specific for the transport of glucose. When these cells need to take up glucose, the vesicles fuse with the cell surface membrane. How does the uptake of glucose occur? Facilitated diffusion 65. Some enzymes are produced in the cells of the pancreas. The enzymes are secreted when required. Which process is used to transport these enzymes out of the cells of the pancreas? Exocytosis 66. Which factors can be changed to affect the rate of facilitated diffusion across a cell surface membrane? 1. The surface R of the membrane 2. The concentration gradient 3. The number of specific protein channels. 67. Which features must always be present for water to move between two solutions by osmosis? 1. Selectively permeable membrane 2. Water potential gradient. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. 68. Solute X is at a higher concentration outside a cell than in the cytoplasm. Which processes may allow solute X to be moved through the cell surface membrane? Active transport, diffusion and facilitated diffusion 69. Which processes can allow transport into or out of a cell? 1. Facilitated diffusion 2. Osmosis 70. Which transport mechanism within a cell can occur in the absence of membranes? Diffusion 71. Which pair of factors is inversely proportional to the rate of diffusion? Distance over which diffusion occurs and size of diffusion molecule 72. Which statement suggests that a membrane protein is involved in active transport? It can only function if mitochondria are supplied with sufficient oxygen. 73. Viskine tubing is often used as a model during experiments to investigate osmosis in plants. What could Viskine tubing be used to represent? Cell surface membrane, represents, cell wall, does not represent, tonoplast, represent. 74. The diagram shows the movement of substance C across a cell surface membrane. Which process is involved in this movement? Exocytosis 75. Which statement describes endocytosis? Movement across a membrane into a cell using a vesicle and requiring energy 76. Which processes use energy in the form of ATP? 1. Endocytosis 2. Exocytosis 77. The formula shows how the diffusion across a cell surface membrane can be calculated. Which row shows the fastest rate of diffusion can be achieved? Surface area available, high, difference in concentration, high, thickness of membrane, low 78. Companion cells use ATP to move hydrogen ions out of the cell and cotransporter proteins to allow hydrogen ions to return with sucrose molecules. Which two processes are involved in this movement? Active transport and facilitated diffusion 79. Which row describes osmosis across a cell surface membrane? Molecule moved uses energy from ATP moves molecule down a concentration gradient solvent incorrect correct. 80. By which process do hydrogen carbonate ions leave red blood cells? Facilitated diffusion 81. Which of the following given processes do enter substances by passive processes? Facilitated diffusion and osmosis 82. Which observations support a link between active transport and ATP production? 1. Active transport occurs only in living cells. 2. The rate of active transport decreases in low oxygen concentrations. 3. Companion cells have large numbers of mitochondria. 83. Which of these processes allow movement in both directions across cell surface membrane? 1. Active transport. 2. Diffusion. 3. Facilitated diffusion. 4. Osmosis 84. Which row is correct for facilitated diffusion of molecules or ions into a cell? ATP not required, movement down the concentration gradient, membrane protein required. Lecture notes for AS level biology by Masha. Cell number 030046100457. 85. Which statements about the proteins in cell surface membranes are correct? 1. They can be involved in active transport. 2. Facilitated diffusion. 3. They can be involved in antigen recognition. 86. Which statements about active transport are always correct? It occurs against the concentration gradient. 87. Antimycin is a chemical that inhibits the function of mitochondria. Which method of transport across the cell surface membrane would be inhibited by antimycin? By active transport and endocytosis. 88. Which descriptions are correct about transport across cell surface membrane? Active process, endocytosis and exocytosis, passive process, diffusion and osmosis. 89. What explains the effect on a red blood cell of being placed into pure water? More water enters the cell than leaves it, so the cell swells and bursts. 90. Which process occurs in a mature red blood cell? Active transport. 91. What supports the view that a membrane protein is involved in active transport? It can only function if mitochondria are supplied with sufficient oxygen. 92. Which feature increase the efficiency of ion uptake by a root hair cell? Many mitochondria in the cell and protein carriers in the cell surface membrane. 93. Which is correct for the facilitated diffusion and active transport? Both require the use of membrane proteins. 94. Which processes that contribute to transport across cell surface membranes are active or passive? Endocytosis and exocytosis are active processes, facilitated diffusion and osmosis are passive processes. 95. What are the features of facilitated diffusions? Uses proteins in membrane, molecules move down a concentration gradient in ATP not used. 96. Which process would allow the movement of large protein molecules out of the cell? Exocytosis. 97. An animal cell and a plant cell are placed in distilled water. The animal cell swells and bursts while the plant cell swells but not bursts. What accounts for this difference? Animal cells have no cell wall. 98. Which process allows the movement of molecules that are too large to enter through a cell surface membrane? Endocytosis. 99. Which processes that move substances across cell surface membrane result in an equilibrium? Diffusion, facilitated diffusion and active transport. 100. A molecule can enter a cell by two different passive processes. Which process would increase the rate at which this molecule enters the cell? Facilitated diffusion. 101. Which statement describes why a large animal has had to evolve a transport system? Diffusion occurs slowly over long distances. 102. Which of the following ways of moving substances across cell surface membrane allows movement in both directions? Active transport, diffusion, facilitated diffusion and osmosis. 103. An indicator mixed with agar forms a pink color. The pink color becomes colorless when put in acid. Blocks of pink colored are cut to different sizes and put in acid. All other variables are kept constant. Which block becomes colorless most quickly? 3 mm x 30 mm x 30 mm. 104. What happens to the surface area to volume ratio of a cube when the length of each side is doubled? The ratio decreases by half. 105. Blocks of agar are stained with a pH indicator and used to investigate the diffusion of an acid solution. Which block would change the color the fastest? 1 mm x 0.25 mm x 0.25 mm. 106. The indicator cresol red changes from red to yellow when put into an acid. Some blocks of agar containing cresol red were cut to different sizes and put in an acid. All. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. Other variables were kept constant. The blocks were measured in mm. Which block becomes completely yellow most quickly? 3 by 30 by 30. 107. A plant cell with a water potential of -600 kilopascals was placed in a solution with a water potential -410 kilopascals for 10 minutes. Which row is correct? Net movement of water into cell, water potential of cell becomes higher and the cell becomes turgid. 108. Raisins are dried fruit that contain high concentration of sugar. Which row is correct when raisins are first put into water? Water potential in the raisins compared to surrounding water is more negative and water moves into the raisins. 109. Equal-sized potatoes pieces were placed into a test tube and covered with the sucrose solution. The test tube was left for 30 minutes. All other variables were controlled. After 30 minutes the potato piece had not changed in size. Which can be concluded from this result? The water potential is the same in the potato and in the sucrose solution, and there is no net movement of water into or out of the potato. 110. Some epidermal cells from well-watered plant are placed in 3 solutions which have different water potentials. Which row correctly shows the state of the plant cells in each of the solutions? Water potential of solution lower than cells, plasmolyzed, water potential of solution equal to cells, turgid, and water potential of solution higher than cells, turgid. 111. What could happen to a typical bacterium when it is placed in surroundings which have a less negative water potential than that inside the cell? There will be a net movement of water into the bacterium. 112. What happens to an animal cell when it is placed in a solution with a more negative water potential? It loses water by osmosis and shrinks. 113. Many freshwater animals possess vacuole which contract regularly, expelling excess water. Why do plants living in fresh water not require such vacuoles? The water potential is the same inside and outside the plant cells. 114. What is a role of essential fatty acids in the body? As part of phospholipids in cell membranes. 115. Which adaptation would increase the efficiency of active transport of carbohydrates from a plant cell? Large surface area of cell surface membrane. 116. A plant cell is placed in a solution with the less negative, higher, water potential than the cell contents. Which change occurs in the cell and what causes the change? Cell becomes more turgid and water diffuses into the cell. 117. Which part of phospholipid molecule makes up the most of the thickness of a cell surface membrane? Hydrocarbon chains. 118. Red blood cells were placed in a solution of sodium chloride with a water potential higher, less negative, than the cell contents. Hemoglobin was released from the cells. By what process was hemoglobin released? Lysis of cell 119. In an animal cell, which process is dependent upon cell surface area and which process dependent upon cell volume? Cell surface area, hormones detected, cell volume, carbon dioxide produced. 120. The epithelial cells of people with cystic fibrosis have a defect in the structure of the cell surface membrane. The ability of the cell to transport chloride ions out of the cell is affected. Which membrane component is involved? Protein 121. Some viruses are able to bind to the cell surface membrane before entering the host cells. Which sequence of events will lead to a virus invading a cell? Binding to a protein receptor followed by endocytosis. 122. Which statement about the fluid mosaic model of membrane is correct? The more unsaturated fatty acid tails of the phospholipid, the more fluid the membrane. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. 5. The mitotic cell cycle. When the body cells were in size, they divided into two cells. Nuclear division occurs first as karyokinesis followed by division of the cytoplasm called cytokinesis. This mitotic